And welcome to our final lecture presentation in our Chapter 2 lecture series. The topic here is our fourth and final macromolecule, the nucleic acids. Now, nucleic acids are composed of the monomer nucleotide, and nucleotides are composed of a 5-carbon sugar with a phosphate group attached and a nitrogenous base attached to that. So this is a nice little simplified drawing of a nucleotide. Now notice that you have a nitrogenous base attached to the sugar, and we have actually 5 possible nitrogenous bases. And these are going to be abbreviated with the first letter of each of these names. So we have adenine, thymine, uracil, cytosine, and guanine. Now, when we talk about nucleic acids, there are actually 3 types of nucleic acids that we'll study. DNA and RNA are studied together. These are our hereditary molecules. Essentially, these are the molecules that are responsible for directing the formation of proteins. So we've already talked about DNA as having the instructions or code to tell the cell how to make the protein, and we will be looking in the future at the role of RNA in protein synthesis. And then the third type of nucleic acid is the ATP molecule, which is our energy molecule. Now, we've talked about carbohydrates as providing 60% of the energy for this cell, and 40% of energy is coming from fatty acids. So we've talked about carbohydrates and lipids as being sources of energy. When we say that the carbohydrate and the lipid is used to make energy, the energy that we're making here is ATP molecule. Let's look at each of these in a little bit more detail, starting with our DNA molecule, which stands for deoxyribose nucleic acid. Now, this is a nucleic acid, and it tells us that in the name. The reason it is called a deoxyribose nucleic acid is this is describing the type of 5-carbon sugar found in this molecule. Now, when we look at a DNA molecule, you can sort of deconstruct it and think about it like a ladder, where the sides of the ladder are sugar and phosphate backbone, and then the middle piece, the rungs of the ladder, are those nitrogenous bases, which again, we're abbreviating those bases. So I have listed here my adenine, thymine, cytosine, and guanine. Now, going back to the previous slide, it's a double-stranded molecule. So we're going to have sugar phosphate on each of the sides of the ladder, and then we're going to have actually pairs of nitrogenous bases in the middle of the ladder. Now, the pairing is happening based on the rules of complementary base pairing. So what we see is that A and T pair together, and you can use this memory device. The apples grow on trees. So if we look back over here at our DNA molecule, where we had an adenine across from it would pair a thymine, where we have a thymine across from that would pair an adenine. And then you can see here, my artistic abilities are highly limited. Your memorization device for cytosine pairing with guanine is that the car is in the garage, so C pairs with G. So looking back at our little drawing of our DNA molecule, where you had a C, you're going to place a G across from that. Where you have a G, you're going to place a C. So adenine and thymine always pair together in a DNA molecule, and cytosine and guanine always pair together. So our DNA molecule is called DNA for the deoxyribose, so the type of sugar that it has. It's a double-stranded double helix. So I've drawn it previously like that ladder, but make sure you're twisting that ladder into a helix shape. And then we've mentioned that the nitrogenous bases are adenine and thymine, which pair together, or cytosine and guanine, which pair together. Your DNA molecule is going to be located in the nucleus of the cell, which we'll be studying a little bit more in the future. And the job of the DNA is really to be the recipe, to have the instructions, to have the code for how to make the proteins or for protein synthesis. This is your hereditary molecule. This is the molecule that you're passing on to your offspring. This is the molecule that you got half of your DNA from your mom and half of your DNA from your dad. The Rna molecule, the R here stands for ribose. So Rna molecule has a different 5-carbon sugar, ribose, compared to the DNA molecule. Another important difference is that it's going to be single-stranded, but it still has a nucleotide monomer, so it still has the sugar phosphate backbone, and it still has nitrogenous bases, but here we're going to see that the RNA strand will have a uracil and no thymine. So we have adenine, we have uracil, we have cytosine, and we have guanine. So make sure you pick up on that difference. There are 3 different types of RNA molecules in the cell. We have messenger RNA, which is abbreviated little m RNA. We have transfer RNA, which is abbreviated little t RNA. And we have ribosomal RNA, which is abbreviated with a little r RNA. Now, these 3 RNAs are all also involved in protein synthesis, and we'll be talking about this process in a lot of detail in the future. So we'll be talking about the role that each of these play later on. The 3rd type of nucleic acid to talk about is the adenosine triphosphate, and this is abbreviated as ATP. Now, adenosine triphosphate is a nucleic acid, so it does have a nucleotide as its monomer. Nucleotides, remember, consist of 3 things: 5-carbon sugar, a nitrogenous base, and a phosphate group. And so we see that here. The term adenosine is referring to collectively the 5-carbon sugar and the adenine, which is the nitrogenous base. And then the triphosphate tells us we don't just have 1 phosphate group here, we have 3. Now, your ATP molecule is your energy molecule, and so we're going to be talking about, a process that requires ATP means it requires energy. A process that makes ATP is a process that's making energy for use in the cell. We have already talked about the cell using carbohydrates or lipids, specifically glucose and fatty acids, to be our sources of energy. So what we will see is that the cell will break glucose down in order to make ATP. The cell can also break down fatty acids in order to make ATP. These are detailed, complex processes, and we will be studying these in the future of the class. So we'll be talking about the process specifically of how the cell is going to break down a glucose molecule in order to produce ATP. And really, you need to just start thinking ATP and energy as being, kind of the same thing. Here is our drawing for our ATP molecule. So remember, it's a nucleic acid, and we can see it has the 3 components. It has adenine for the nitrogenous base, it has ribose as the 5-carbon sugar, and it has actually 3 phosphate groups instead of 1. Now notice I put a little red squiggly bond in between those phosphate groups 2 and 3, and the reason for that is this is where all of that energy is stored. Our phosphate groups have a negative charge assigned to them. And remember, we've been saying opposite charges attract, which means like charges repel. And so basically what's happening here is you're forcing these negatively charged phosphate groups to be close together. So think about if you have 2 magnets and you're trying to bring together those opposite ends of the magnets and how they don't really want to touch each other, and you're bringing them close together. And that's what you're doing here with the phosphate group. So you're bringing these negatively charged groups as close together as possible, and you're holding them in place, and they don't want to be close to each other. So we're going to think about those bonds as our high energy bonds. These are our potential energy bonds, our stored energy. When you hydrolyze, when you break down your ATP, you're going to be left with an ADP, adenosine diphosphate, a phosphate group, and a bunch of energy that's released. So if you had those negatively charged phosphates close together and they don't want to be close together and you allow one to break off, now you get this release of kinetic energy that's available for the cell for work. And that, ladies and gentlemen, is the end of chapter 2. Yay!